objectives are to popularize science, create a scientific outlook, organize lectures and seminars. Since last 54 years, every Wednesday and July 1 month at 6 o'clock, talks are conducted on interesting topics delivered by various eminent scholars. Today, 2725th talk on a new language for biology in the 21st century. Are we equipped to speak it? It is going to be shortly delivered by Professor Satyajit, Director, National Center for Biological Sciences and Institute of Science and Biology and Regenerative Medicine. On behalf of Bangalore Science Forum and all of us here, I extend a very warm welcome to Professor Satyajit studied chemistry at the Indian Institute of Technology, Bombay. He was awarded his PhD in Life Sciences from the Rockefeller University, New York. He worked as a postdoctoral fellow at Columbia University, where he developed tools to study the trafficking of mammalian lipids and GPI-anchored proteins in mammalian cell using quantitative fluorescence microscopy. Professor Mayer started his laboratory at NCBS in 1995 after the completion of his postdoctoral research at Columbia University. The broad aim of Professor Mayer's laboratory is to determine how control endocytosis imposes on many development programs in tissue morphogenesis. Professor Mayer is a recipient of several national and international awards and honors, such as elected as a Foreign Associate of U.S. National Academy of Sciences 2015, distinguished alumnus of IIT Bombay, elected EMBO Fellows 2013, Infosys Prize for Life Sciences 2012, the World Academy of Sciences Prize in Biology 2010, EMBO Global Lecture 2010, J.C. Bose Fellowship DST 2006-2011, Shanti Swarup Bhatnagar Award in 2003, Swarna Jayanti Fellowship DST 2003-2008, Wellcome Trust International Senior Research Fellow 1999-2004, and the Rockefeller Oxford Student Exchange Program recipient 1988, and many more. This lecture is organized in memory of Srimati Yogamula and Sri R. Adinarayana. We are happy to have his son, Dr. A. Srinivasan here. I extend a warm welcome to you, sir. With this brief introduction, once again, I extend a warm welcome to Satyajit Mayer. Please continue.
While the mic warms up, thank you very much for inviting me to speak at the, I think it's 39th Bangalore Science Forum Science Festival. It's, as always, I'm pretty amazed that there's such enthusiasm for bringing on people from far and wide to talk about contemporary science in this audience. It's also impressive to see both young and old come and take part in these discussions every year. I'm a little embarrassed because I actually recognize people from the last few times that I've given these talks. use the mic.
Okay.
Yeah, so I'm a little embarrassed with the familiar faces because some of them are going to see a repetition of a few things that I talked about last year. But perhaps they may not remember it because this festival is so big and has so many people talking about so many different things that if they've come to many of them, like the two of you in front, they've come to many of these talks, perhaps you will forgive my repetition in the interest of making some of the points that I had to make last year a little more explicit and a little clearer this time. At least it's been a year in its telling, so the telling might have improved a little bit. At least that's what I hope. So I do want to talk about the new language of biology in the 21st century. And we have amidst us also a historian of biological science, in fact, molecular biology, Simon sitting there. So he might have a lot of comments to raise, and perhaps please feel free to raise them, anyone, during the course of this lecture. So what I am going to do is outline in the following slide. Yeah, so I do want to talk about a little bit about what I did mention last year. Sorry. Sorry about that. I do want to talk a little bit about what I had mentioned last year, which is the shifting geography of biology, and of course the shifting language of biology. There is incredible opportunity in this shifting geography, especially for us in India or in Asia. There's tremendous opportunity for basic research if we can actually begin to grasp that, and I want to say a few words about what aspects of that basic research can we engage with. I do want to make a big plug for serendipity and discovery, and I'm going to tell you a story that has begun to emerge recently about something that I think we all should be thinking about, both as scientists and as citizens and as people part of a larger society. And finally, I'm going to end up with telling you something about this new language for biology and the challenges that confronts us, those of us who both want to study biology and those of us who would like to teach biology. I mean, it's a fabulous opportunity. So let me start by saying that a new fabric is being woven for the biological sciences in the 21st century, with a general increase in the emphasis on biology globally. The major geographical enhancement of this emphasis in biological sciences is actually happening in the Asian continent, or the Asian configuration. That I would say both in China, in India, and in South Asia, there is a huge increase in activity in the biological sciences. In some sense, there's a saturation of growth in doing science in the West. And it's a huge opportunity for us in India, where our economies are growing and where the expenditure on science is actually not growing in step with the economy. That's I think a little sad. I'm going to touch on some of the numbers, in fact, right now. So if you look at the funding for research in Asia, the 20 countries in Asia, including India, have a positive slope. In the Americas, this includes all countries in the Americas. In America, of course, the North America is the biggest contributor to that. The line is almost flat, and it sort of declines now in 2016. I don't have the details there. And compared to the rest of the world, where again the line is flat. So in fact, the only place where growth in research and research funding is taking place is in India. There are many different priorities amongst, but you see, if you look at the global distribution of the research bubble, if you will, funding versus people engaged in funding. I'm sorry that these slides may not be that visible at the back, but
One side you can
pardon?
One side you
I can expand it on slide. Okay, I think the general point I wanted to make is not about the details, but about where we lie. So the bubble, the big circle, represents the total bulk, total amount of money that a given country is putting into the size of the bubble is the size of the total expenditure of research. For example, blue is United States. China is down here. China is one of the biggest compared to the United States. India is here. And as a function of GDP, India is less than 1%. China is getting close to 2%. The US and all the other growing economies, but all the other developed economies of the world are between 2 and 3% of the total GDP. And on the x-axis, we have the number of people engaged in research with this per million population. So it's a per million population number. So you have India really at the very bottom. Number of people engaged in research is small, and the size of the amount of research going on is small, and also the fraction of GDP is very small. Now, this is not a good thing for a country of our size, of our scale, and with a growing economy. We are pretty much down there. But there's one good news, is that the distribution of research in India is pretty equitable between basic research, between applied research, and development. So development would mean technology development. So basic research is sort of the brown and light blue, and dark blue is applied and development. Development would mean directly product development. South Korea, as you can see, is got a similar emphasis. So the South Korean bubble is actually much bigger than ours. In South Korea, the tiny country spends more money than we do on research in general. And Russia, of course, is such a distribution. Now, regardless, I think it's very important that we maintain this emphasis on basic research as the economy grows and as the expenditure on science grows. But the bad news is that India's support for science has been, as I said, below 1% of its GDP. It's been flat for many, many years now, and it continues to remain flat even today, 2016, and in fact, it barely keeps up with the inflation. And that's not a good sign, because I think we should be somewhere here by now, today. And I think this really reflects the capacity of, if you will, of scientists, perhaps like me and others, to communicate the need for emphasis on science to our political masters, whoever they are. But also it does reflect the willingness of our political class to listen to some of the communications that they receive from us. I think it's going to be an uphill battle, but we have to be able to take the argument that emphasis on science has to grow. And I think there are a number of reasons for that. And I'm going to go back to what I really love, and that's biology. I'll tell you a story about what happened to biology and the discipline of biology from somewhat historical context. So biology in general has seen, biology today is a consequence of actually many sort of almost tectonic shifts in thinking about the discipline. In contemporary sense, biology was, before the 1950s, a predominantly descriptive type of science with a few driving concepts. So people were descriptive and also, had a huge emphasis on organizing and classifying the world of the living, and providing descriptions of what was there, with a few broadly unifying perspectives that provided a core basis to think about a living system. One core perspective was the notion of evolution, and the fact that every living being was made up of a cell, of a living cell. So there was the idea of the cell theory and evolution, and evolution combined with the laws of genetics provided a very good way to think about the world around us. Of course, this had no need, if you will, for any of the natural sciences. This had no need for any engagement with any of the other sciences to be almost a self-contained bubble. And I think they're fantastic examples of how the idea of evolution, and the notions of evolution, and the laws of heredity came from two people. I mean, they were theologists in their training in the beginning. Charles Darwin and Gregor Mendel, who began to think about evolution from a perspective of how you describe the wondrous beings of God's creation on earth, and then by sheer application of mind and reason came to these very, very general laws that effectively apply to every living system on this planet. The law of evolution and law of heredity. Ironically, while these two people, Darwin and Mendel, were living about the same time, one man was a celebrated scholar, and the other was a poor monk who actually died without even knowing that his work had been read by anyone. In fact, until he died, there were maybe 4 citations to his own work. But then, of course, once the connection between evolution and heredity were discovered and the principles of heredity were thought through, there was a huge, rediscovery of Mendel that took place in the early 1900s. But this, again, was again in the context of a very confined, dialogue about biology. And about the same, in the late in the mid, or I should say, the early 1900s, there was already the inquisitiveness of the natural scientists, physicists and chemists, to ask, we have these particles that are responsible for heredity. They're called genes. What is their physical basis, or do they have a physical basis? And there was already, huge churning amongst a number of physicists and chemists who began to see the world as a world that could be explained by the physical laws. The laws of Newton could explain not only, the fact that traffic moves slowly in Bangalore, but that planets move around each other, and if you trip, you will fall. I mean, this very powerful explanation from physical sciences provided, I think, a sense that to physicists and chemists that one could, in principle, if he knew enough, explain biology as well. And Francis Crick and some of those people who began to think about the DNA, but began to think about DNA. Harvey and Chase, who actually discovered that the unit of transmission of information from one organism to another was DNA. Their experiments are legend, and I'm not going to talk to you about them, but they did, in fact, produce what you will, in some sense, the idea that you could explore biology from a reductionist and mechanistic perspective. These two, I would say, programs in biology, one of being able to describe and explain from broad principles, and the other being able to go into a chemical or physical basis for life, were products of different eras, and they were products of different ways of thinking. One of them was from the sort of European and the Renaissance of the Enlightenment period. The other was a more heavily driven by the American sort of innovative spirit, where it was, in fact, I think, a largely American revolution, a molecular biology revolution that biology has experienced in the last 50 years, 1950 to 2000, till to the description or describing of the human genome, has been a largely American revolution in understanding the elements or the chemical basis of the living system. So, but none of this would have been, the reason I'm saying these two things about emphasis on basic research and giving you a bit of this history, is to say that the American Revolution was predicated on the backs of an extraordinary expansion in the research enterprise in America at that time. After the Second World War, one of the chief architects of perhaps the worst thing that we know from the atomic bomb, the chief architects of the Manhattan Project, Vannevar Bush, no relationship of the Bush family, but Vannevar Bush, he was an advisor or the director of the Office of Scientific Research and Development and advisor to President Roosevelt. He wrote this incredibly influential piece called Science, the Endless Frontier. It was a policy document saying that, even though he had been the architect of this huge mega project, something that's going to sustain the world, and definitely America and its economy, was going to be basic research. Research that uncovers new things that nobody's even thought about before. And this document simply, it was, I think, a very brilliantly argued document that forced the American president at that time, who had all these other demands on the money that the Americans had at their disposal, for the war on cancer, the war on infections, the war on so many other things. But it forced the American president to sit back and say, well, I think this is a fantastic opportunity to build for the future. So this document, I think, Science, the Endless Frontier, argues that unless you are going to invest in basic research, there will be nothing to sustain you beyond a few steps that you might take. In fact, he argues from the building of the atomic bomb that unless there was the research of the early part of the 1900s in physics and chemistry of understanding nuclear energy, there would never have been the atomic bomb, and that was certainly not the inclination at that time. So, unfortunately, we need such champions who can convince our governments that investing in basic research is important. And I know I'm speaking to the converted here, but I want to tell you what opportunities we have, and if ever you have an opportunity to talk to some of our erstwhile politicians, it may be worth giving them some of these arguments. So I'm going to repeat a few things that I had said last year about the shifting, what will be the implication of this shifting geography of building up basic research, assuming that happens in our neighborhood. Well, what I'm showing you here is a map of consanguinity in our human populations across the globe. It's a map that was derived from work from Littles in 2007, published in the Proceedings of the National Academy of Sciences in the US, where he shows that there are some pockets where consanguinity, which is kin marriages, marriages between cousins, or first cousins, second cousins, but marriages within families is extremely high. So you have some parts of Africa, some parts of the Middle East, West Asia, Pakistan, and South India. South India is also a very, and this is 2007, and this today is less, so the darker colors are higher consanguineous marriages, and the very dark color is greater than 50%, means every second marriage is consanguineous. And the numbers in South India average around 30%. So every third marriage or fourth marriage is a consanguineous marriage. And with the emphasis on basic research in India, we have the opportunity to deal with the consequences of this consanguineous marriages. I mean, it's a strange statement to make, but the consequences of consanguinity, the social and economic costs are extremely high because of the morbidity associated with consanguineous marriages when there are mutant or defective alleles that are coming together. And very often one sees an enhancement of morbidity or diseases, maternal and prenatal factors, congenital defects, single gene disorders, and specific infection susceptibilities, as well as adult onset dementias, and getting enhanced with this extent of consanguinity, simply because recessive genes. And this is something Mendel knew. Recessive genes, when they come together, can give rise to phenotypes that are manifest often as disease. Now that's the situation that we have on hand. But in fact, given the incredible genetic diversity of our population, and there's a beautiful study that came out a few days ago again in the Proceedings of the National Academy of Sciences, from Partha Chatterjee in the National Institute for Biomedical Genetics in Kolkata, a study where he says that the Indian population at large, the South Indian in particular, has been peopled by many different sources, and yet maintains an element of sort of interbreeding, which is, for a geneticist, for a human geneticist, it's an absolutely incredible opportunity. So, an incredible opportunity to uncover the traits, both physical and pathological, traits for which the opportunity doesn't exist anywhere else. And if you construct that with a strong research base within this geography, the opportunities for understanding diseases that could affect not just us but the whole world are incredible. And I'm going to tell you how, as an institution, we have taken some steps in trying to leverage this, if you want to call it this advantage that we have. I mean, I shouldn't call it an advantage, but for understanding disease, it certainly is an advantage. It's not an advantage if you are subject to some of the morbid issues that the consanguinity raises. Okay. So basically, today, one can take people who are affected by a particular disease and take blood samples from individuals and convert blood cells into what we would call induced pluripotent cells, or iPS cells, or stem cells. But those stem cells, if they are coming from backgrounds that have disease associated with them, can serve as models to understand the disease condition in the animal, in the human. So the stem cells that we get from the human can be then differentiated into different tissues, into liver, brain, heart. When I say tissues, I also mean cells and then building up of tissues. And therein lies a huge opportunity, which I will sort of come to. And those can then serve the healthy cells from healthy people can serve as controls, and the cells that come from people who have the disease can serve as disease models. And then their understanding for basic cellular tissue mechanisms becomes wide open and is available to study in a dish rather than in a clinic where it becomes far more difficult to explore the fundamental mechanisms behind what has gone wrong. And then therapeutic evaluation and interventions become far more easier to think about. But it's very important that one is engaging with serious clinical diagnosis, have clinical scientists, people who understand human genetics, familial diseases, and very, very strong case histories recorded. And one such example is, where all these things have come together is a study that we have just begun at the National Center for Biological Sciences and in STEM, in collaboration with the National Institute for Mental Health and Neurosciences. It's a study that has been undertaken by three of my colleagues, Mahendra Rao at STEM Raghu at NCBS, and Satyajit Jain leading some of this work at NIMHANS, but of course with a number of people who are equally collaborative and equal footing along with them. These are, and the work of this study has been recognized by the Department of Biotechnology, and the Pratiksha Trust, a private foundation initiated by Chris Gopalakrishnan of the Infosys, former Infosys director, founder. So the study is the following, that clinicians at NIMHANS have collected clinical cohorts extending about 30 years of people with serious neuropsychiatric disorders. And these people with serious neuropsychiatric disorders have familial, carriers of familial disease, many generations. I mean, it's and that information, unfortunately, for the families have been preserved because of this interbreeding prevalent in society. But given that in today's snapshot, this configuration allows one to now actually, because these case histories have been studied and documented so carefully, allows one to go back and explore the fundamental mechanisms behind some of these neuropsychiatric disorders that would never have been possible anywhere in the world. So we have for the first time an opportunity to take what we have at our doorsteps and be able to explore it. The information that we're going to gather from a study like this, where we take information from patients, create the stem cell models from the blood samples collected from these patients, and then try and understand the disturbances that have taken place in the brain cells of the patients who have had such a disorder is just absolutely unimaginable. I mean, it's really an extraordinary opportunity that unless all these three elements, unless the National Institute of Biological Science, the Institute for Stem Cell Research, and NIMHANS were actually were of the capability and caliber to do some of these, stitch some of these ideas together and work together, none of this would have happened, would have come together on the same platform. And 5 years from now, or 10 years from now, this possibility would be lost simply because the evidence in the well, first of all of us are aging. Our clinicians who have got all this information would have retired and lost access to their patients and to their records. And what is even more likely is that the evidence in the genetic background of people would be dissipated because consanguinity is, of course, on the decline. And in the next 2 generations, the extent of consanguinity is actually going to disappear quite substantially. going to catch up or going to become somewhere similar to the rest of the world. So this, creates, as I would say, this possibility that we look at our own populations and ask, what is it that we can do that in fact creates an understanding of diseases that have never been understood before. So when I talk about neuropsychiatric disorders, one could put any other complex disorder in this place. One could put, the extraordinarily afflictive and chronic disease of diabetes that is affecting our country today. And if we had clinical cohorts and people willing to take this on, this whole program on, this would be an extraordinary opportunity today. Unfortunately, because our basic science and basic research hasn't been built up to the extent that can take advantage of what we can produce from here, we will have to and we must engage with our colleagues across the world so that we can share this resource of information to understand better the problems that affect us. So it could be diabetes, it could be cardiac disease, or any of the chronic disease drivers that exist in our midst today can be approached by a similar kind of program and platform. But this will be a engagement of basic exploration. We'll find new things, and of course of potential outcome in the long term. And it cannot be that one will see a new drug emerge in the next few weeks or next few years. But definitely a new knowledge and new information will emerge that will inform how we can begin to tackle some of these diseases that have been extant for thousands of years. So the shifting geography of basic research in biology, and couple that with the incredible human genetic variation in our doorstep, brings about a massive opportunity. But it does say that we will need to change the way we think about teaching biology, about being able to teach biology, because I think what I didn't mention here was the need of extraordinarily deep sequencing and genetic analysis of the genomes of individuals who are affected and not. So we will need to create, and the amount of data that will come out from such a study will be a challenge just to even put together in one place, digital information to put together in one place, let alone analyze. So we need people to be able to think about those challenges of how we begin to mine this big data that is going to come out from just the extraction of genomic data or genomic information from the patients that we are going to look at. So the very teaching of the life sciences, the understanding of genetics and genomics, how organisms and organs and tissues develop, the cellular mechanisms that are contributing to the diseases, the basic cellular tissue mechanisms that are disrupted in disease models or in diseases, and how do you begin to evolve new strategies using such a configuration. And I think this is where biology, if it is connecting to society, is going to go. This is sort of the way that biology will connect to medicine, and that's something that is happening in front of our noses, and I think it's a great opportunity to take that into account. And I'm a basic cellular biologist. I'm not a clinician. I'm not actually somebody who's particularly interested in disease. I shouldn't say that to funding agencies, but I really want to understand principles behind biological context. And I think there is a very happy marriage possible between being able to get a knowledge about diseases and disease-causing agents and fundamental biology. And this is because all of biology is related. Anything that we have in our genome has come from an evolutionary cauldron that has all kinds of animals. It has bacteria, plants, fruit flies, So if we can understand, and many fundamental mechanisms of biology may not be observed if we are only going to look at the human or tissue from human beings, because they may not exemplify the specific process that biology engages in, that a biological process engages in, to the extent that it is available for study. And let me explain that a little bit. So I did mention that we can understand and learn a lot from human genetic variation, but in order to understand the why and how things happen, as a consequence of this human genetic variation, we do need to have model organisms. We do need to have organisms that we can quickly manipulate and work with and ask how these variations and changes can affect developmental plans, brain configuration, and body plans. And this is something that, as biologists, we are full aware of. And I think what we are also aware of is the acute paucity of model organisms that can exemplify specific programs in biology. And here again is again a huge opportunity which I'm going to mention after I tell you why I think it is that we need to look at certain things. So, for example, by studying Baker's yeast and fission yeast, Paul Nurse and his colleagues discovered the cell cycle, discovered how a cell grows and then goes into a period of synthesis of the new components, then divides and then grows again. That process is precisely what goes wrong in many cancers. So by studying Baker's yeast, we're able to understand how you can actually address many cancers. Actually, many cancer therapeutics today are based against molecules that were identified in the screens for how Baker's yeast grows. Because we understood how Baker's yeast grows, and because it does it all the time, and it's a eukaryotic system, the rules that apply to Baker's yeast would apply to our own cells. I mean, I'm being a bit, I'm stretching the argument a little bit, but the fundamental rules apply. And there are details that are different. But if you know what the fundamental rules are, we can bring those rules to look at systems that are perturbed in our context. Chromosome ends, again, was discovered in the organism called tetrahymena, that many of you may not even have heard of, but the tetrahymena has enormous number of chromosome ends because it has enormous number of chromosomes. And the discovery of telomeres in tetrahymena is again very fundamental to our being able to understand why cells grow immortal. When the telomere shrinks, a cell actually enters into a senescent cycle. And when the telomere is maintained at a certain length, the cells can continue to grow and divide. Many cancers, again, disrupt that mechanism so that they bypass this telomere checkpoint, if you will. And understanding of that came by studying this single-cell creature called tetrahymena. Trypanosomas are sleeping sickness parasites. Also, we've learned a lot about basic fundamental biology from trypanosomes. And Drosophila has told us how body plans are laid down. The precise way that the body plan is laid down in a Drosophila and the orchestration of genes and its programming, it's almost, I mean, if you didn't know any better, what the final outcome were, if you were look at that orchestration, you would see that that's very similar to what happens when the body plan is laid down in a human. So the connections due to evolution and common evolution are an extraordinary opportunity again in biology. But we need more model systems to engage and discover many more things. A great example of this is something called the CRISPR technology, which I don't know if many of you have heard about this, but this is something that is going to, in some sense, is a technology that's come into biology today. again, from a serendipitous engagement with the way research and science is done. It's a technology that is about genome editing. Today, it is actually incredibly facile using this CRISPR technology to edit the genome of any living organism, including us. One can change single nucleotides, stretch the DNA of a yeast or a human embryo with the same ease. This technology has completely transformed and revolutionized the ability to modify the genome of any living organism today. It's exactly what, basically one can write a new code into the genome today. And this is, incredibly powerful, and I think it's very important that people understand that this is something that can be done today. We need to begin to understand how, as a society, as a group of human beings, we are going to deal with this. We need to understand, have discussions about the societal implications of this technology. And of course, since it's so revolutionary, we need to have ethical and reasoned debates about the use of such a technology. And it's perhaps good to start when the technology is new and is going to become available for use very, very soon. So it's good to be able to appreciate some of these tools and technologies that are coming into biology as we speak. But this technology, powerful though it may be now, didn't start out as something that was what the initial discoverers designed or wanted to happen. It actually is a fascinating story, and I would encourage people to read about how this technology came to be. But I'll give you a brief glimpse of what I can in the. So, again, the maps are a bit odd, but let me just say that initially, the green dots that you see here are little dots on the Mediterranean coast in Europe, where two groups, one in France and one near Spain, were working on how bacteria are able to fight, or how bacteria accumulate very funny pieces of DNA inside their genomes, repetitive sequences of DNA inside the genome. And they were just obsessed with, why do bacteria, E. coli, things that live on our skin and elsewhere, all of them have this repetitive sequences of DNA inside them. And how do bacteria actually begin to collect such sequences? And then, of course, the question is why. And this reminds me of a saying from Wittgenstein: how a small thought it takes to fill someone's whole life. This is a very small thought. Why do you have so many repetitive sequences of DNA inside the genome of a bacteria? And then they discovered that those repetitive sequences of DNA in that bacteria are actually similar to many of the viruses that the bacteria encounters, the bacteriophages that the bacteria encounters. And then the whole, the penny dropped, if you will. And they discovered that the bacteria had created an adaptive mechanism to deal with bacteriophages that come from the outside. These little sequences of DNA are then able to guide, they're called guide RNAs, they produce guide RNAs. You can guide in a complex that chews up the genome of those precise parts of the genome of the virus and renders the virus inactive. So it was from this very simple sort of looking, just without any, a smaller thought, without even asking, what are the big implications, just looking and being curious, that this technology was discovered, where you can now adaptively change the DNA of any organism. So today we can take that piece of RNA that corresponds to a segment of a genome of any one of us and put the enzyme that is needed for the cleaving of that DNA into any cell of our body or any cell of any organism, and actually change the genome of the organism. So that, and the study has now become one of the CRISPR story has become one of an extraordinary, as you could imagine, a battle of sorts of patents and litigation and priority and all the nasty things that that accompany technology coming into being. But it's going to be very important for us to take this on board and think about it. Again, we need to have scientists and enlightened people, the Bangalore Science Forum being one where such discussions can happen and one can think about the implications of the technology that's at our doorstep. Actually, there's a very interesting article by Eric Lander, who's the head of the Broad Institute, where they do a lot of sequencing and understanding the meaning of these sequences. And he writes, and I want to read, the narrative underscores that scientific breakthroughs are rarely eureka movements. They are typically ensemble acts played out over a decade or more, or millennia or more sometimes, in which the cast becomes part of something greater than what any one of them could have done alone. It's actually a wonderful lesson for the general public as well as the young person contemplating a life in science, that thinking about something deeply, even if it is a small thing, is not a bad thing. I mean, that's precisely what Mendel did when he was going about breeding his pea plants. I mean, it was precisely that. How do you take a small idea and then explore it to its fullest? And I think that requires a support from society, which is broad, open-minded, and able to support, able to create an environment where dialogue and discussion can take place about the outcomes of such an activity as well. Okay. And of course, it needs institutional mechanisms to support activities. It needs places where one can go and do things. And I want to tell you today about NCBS and two people, Krishnan and NCBS. And maybe I'll skip NCBS, but I want to tell you a little bit about Krishnan, who was a dear colleague of mine who sadly died a couple of years ago. But he left us a tremendous legacy about how one might take doing science as a creative act in itself as a human activity. Ubaid Siddiqui, the founder of NCBS, would be very proud that NCBS today is celebrating 25 years, not today, but this year we'll be celebrating 25 years, and has spawned many creative institutions and creative new institutions. So, and Ubaid was of the mind that as long as you encourage excellence and let individuals explore what their minds and hearts want to explore, you will find good things happening. And I think, in some sense, he was right. It's very hard for funding agencies to see that. But I think you really have to think about building institutional structures that will allow that activity to take place. And Ubaid's prescience, goes into the building of NCBS as a place where you can study molecules in biological context to ecosystems, all under the same roof, and at the same time build programs that are connecting institutions like the National Institute for Mental Health and Neuroscience with a fairly embedded, deeply clinically motivated activity that I mentioned a little while ago. And I think that's the kind of space that one needs to be thinking about if you're thinking about institutions that will support science. But Krishnan, it was a particular case where he himself is an electrical engineer. And I'm sorry I'm going round and round, but I'm going to come back to this idea of a new language. But I would say Krishnan was one of the first people who began to explore a new language for Krishnan, and I'll explain why. Krishnan was an electrical engineer by training. He went to the BRC training school, and he was destined to go off and make detonators for all the nuclear devices that we have explored. He actually put down a few designs for some of them. But then he realized that he didn't want to spend his life doing that. He was an naturalist at heart. He loved to watch what was happening in nature, and he made amazing observations. So he actually, when he was taken on by Obeid as a complete maverick who could say anything he wanted and do anything he wanted, but would back it up with observation and meticulous, detailed analysis. He actually was quantitative as a biologist, quantitative as a scientist. Quantitation and making measurements was part of his DNA, if you will. And he did a lot of that. He built this device, and it's the device for which we should recognize our understanding of anesthesia. We call it the inebriometer, and it does just that. It makes fruit flies inebriated, because he can put in this device, it's 2 glass columns, and he puts fruit flies on top, and puts a volatile substance that makes the fruit flies anesthetic, or makes the fruit flies go to sleep, anesthetizes the fruit flies. And those fruit flies, which are mutants, escape this column, this sort of distillation column. And the fruit flies that remain at the top and are not collected at the bottom are the ones that he studies and asks what are the mutations that are carried in those flies which prevent them from being anesthetized. So based on this simple analysis, of course this requires a lot of measurements and numbers and all that, allowed him to identify the locus of anesthetics. Never had this been done before. In fact, his work has been again, widely unrecognized, but it is the pioneering work of our understanding of how anesthesia works. Then Krishnan decides that he's going to go and find toxins from cone snails or snails. And I asked him once, why do you want to do that? And he says, snails sit in one place and they move very slowly, but they eat fish that move very quickly. So they must be doing something to the fish that make the fish become part of their meal. And he, of course, discovered that the snail put out toxins that immediately paralyze the fish and make them do all kinds of things, make them tame, make them pets, make them, like farm animal. He discovered many, many ecological kind of configurations that snails actually make the fish do. And this was, again, by simple, observation and thinking about, how could something like this relationship work in nature. So his idea was that simply by observing nature and making quantitative measurements, you could learn a lot and then discover many things that could be of great value to us. So some, not Krishnan, but some others have done similar things and discovered very, very potent painkillers that have come from snail products, snail peptides, that perhaps Balaram may have talked about this, because Krishnan did this work with Balaram. And Balaram did all the analysis for Krishnan and Balaram go back very, very long time. Krishnan happened to be Balaram's first student, but Krishnan was older than Balaram, and he had joined Balaram when Balaram had just started his lab. And Balaram didn't know who was driving the lab, it was Krishnan, not Balaram. Krishnan again was an extraordinary, the charismatic person who really inspired many of us. So, but what he's truly inspired is a gang of people, and I think you heard from Shannon Olson in this room a few weeks ago. But he's inspired a whole host of people, Balaram included, and Rene from ISC and a number of our colleagues from the Agriculture University, to put together a program which we are going to follow in Henry Krishna's great enthusiasm, that we will go out and look at what our neighborhood has in terms of these ecological interactions and identify the language of nature nature, which is chemistry. The language that, I mean, I'm speaking in language where vibrations are impacting your ear, but most of nature speaks in the language of molecules, not in the language of physical perturbation of the air, or and language of molecules, which is in fact creating color, creating smells, creating all kinds of influences, hormones, and that's something that we hope we will discover with this program that we have. Of course, another big component of building a big, robust basic research program in India is our huge traditional medical knowledge. And if we can begin to understand that from such a complex perspective that biology begins to offer today, we have an enormous opportunity to even again for health and disease. Now, again, in the interest of time, I'm going to shift gear a bit and get back to this, and I've been alluding along the way about how biology has been impacted by the different disciplines of the natural sciences. But biology is actually quite unique in the sense that I think it demands a knowledge of the natural sciences, that is, that the natural sciences have just begun to deliver unto us. Unto us, when I say us, I mean biologists, the larger community of biologists, who could be physicists, chemists, mechanical engineers, or anybody. The people who want to study biology are the biologists. And I think the natural sciences as a discipline have begun to deliver unto us principles that have not been available until today. And I think this is something which I think is very, very exciting. So the biological sciences need the natural sciences for its understanding. It is also historical and based on the concept of individual within a population. This is very different from the concept of a molecule in, say, a mole of a substance. Because there is, an individual is not a molecule in a collection of other individuals. Oh, sorry, an individual is not a homogeneous entity in a collection of other individuals in a population. Just like a molecule is a given molecule is identical to another in a collection of molecules of similar kinds. So chemistry differs from biology in that very fundamental aspect. And I think that's a concept that is also beginning to enter into the natural sciences as a way of being able to grapple with this idea. And one fabulous example of our being able to understand the gene as a concept to a material, which is DNA, has already been played out in the early part of this lecture that I started with. But how do we understand how the material, that is the DNA, contributes to the idea that the gene is? And I think that is the crux of the question today. We know every material inside a living cell today. There's very little of a cell that we do not know in terms of its constituents. But we do not know how these materials actually constitute a living system, or even units of a living system in the nature of its organization. But today, the physical sciences and its integration biology are providing us really fundamental new insights into how we can put the chemicals together and create systems that living systems work with. So, for example, here is a molecule of a structure that builds a filament called the actin filament. And this filament, if you just take that molecule and put it in a test tube, it will build a filament. Just because that's what it does. I mean, you take sugar and you can evaporate a bit of it and it will crystallize. Similarly, you can take actin and put it into a test tube and it will build a filament. But in the biological context, the biological context does not allow it to build a filament unless it is completely regulated. So the system builds a network of connected molecules that are preventing this filament from forming unless needed in a given place, given site. And the chemistry by which it actually builds this filament can be fundamentally different from what happens in a test tube. And how it is different and what is happening inside the cell is what we are beginning to understand today. And the rules of that are violating rules of the standard first and second laws of thermodynamics, simply because the system does not obey detailed balance. The system is taking energy to inflict this regulation and creating something completely new that would not have happened in the standard world that we are used to. In a world of equilibrium that we are used to studying, that is completely different from the world that the cell works with. The cell works in a world where energy and ATP is being consumed to effect this kind of a regulatory system, and then create structures that are actually look quite unbelievable, but they are actually today completely understandable if we understand where this energy is being inflicted or injected into the system and how that is being utilized to create or overcome the laws of the first and second laws of thermodynamics. So, we are beginning to put together what I would call mesoscale biology, biology at the micron level, which from a first principles natural science perspective. And this is something that is completely available within our grasp, and many of us are engaged in such an exercise where we can begin to understand how energy, ATP, that is the currency of energy inside any living system, how that is available, how that can put together a very complex structure inside a cell, very different from what would happen in a test tube where none of these things were present. And this is this is where biology is today in terms of this new language. It is beginning to build a theoretical basis from the natural sciences, from modeling, from condensed matter physics, from physical chemistry. It is connecting to engineering as a discipline that allows us to solve problems that are created by construction, literally physical construction. Take computer science and use information theory that computer science gives us to understand how information can be encoded in some of these structures that the biological system builds. Of course, applied mathematics is the language that we use in all these disciplines, and that applied mathematics allows us to make predictive theories that can then be tested in the experiment. And the experimental armory is opened up hugely with the new tools and technologies available for of imaging molecules in a biological system that are now no longer limited by the laws of diffraction. So today, biology imaging has been able to overcome by tricks, for sure, because the diffraction is diffraction, and you can't overcome it. But you can make great tricks with the system to image sub-diffraction systems in living organisms today. So, with the combination of theory and experiment, we're able to do this. And I know I've gone on for very long, and I'm just going to end here by saying, biology, that is the study of animals, organisms, humans, and otherwise, have been so far the purview of molecular and genetic inputs, and that's how we biology describe both cells, tissues, and everything else that go on to make an organism. But we now have the capacity to put in physical, chemical, and mechanical inputs into the very description of a very understanding of how a complex system can be put together. So biology is a hugely interdisciplinary exercise, taking from mathematics, computer science, natural sciences, material sciences, the electrical engineering, civil engineering, and the mechanical engineering. All that these disciplines can offer can be put into the understanding of any biology. I mean, to take any system that you want here. It could be the muscle in your heart, and you need all these different disciplines to understand, the muscle in your heart, or the membrane in your ear that is allowing you to hear what I'm saying. But these shifts in language, and I think the geography I said is an opportunity, but the shifts in language are creating a challenge for many of us, because I think it's important that we have the next generation able to appreciate some of these shifts and be prepared to take them on. Because I think biology is going to be the discipline of this century. It's going to be the discipline of now that will of this century. And we need to be able to take our young people and expose them to the capacity to take this on in a full body manner. Okay. And I'm going to end again with another quote that I had also read out last year, but I think it's even more pertinent today. And it says that in every section of the, and this again is not Weaver Bush's quote, in every section of the entire area where the word science may properly be applied, the limiting factor is the human one. We should have rapid or slow advance in this direction or in that, depending on the number of really first-class men, he wrote men then, I'm writing women now, who are engaged in the work in question. So in the last analysis, the future of science in this country will be determined by our basic educational policies. And I think he put in very appropriately a note of warning. He said it would be a folly to set up a program under which research in the natural sciences and medicine was expanded at the cost of social sciences, humanities, and other studies so essential to national well-being. And I'd say to our this ability to understand how technology or the fruits of such research impact our society. So thank you very much for your attention. Thanks to people with me in this journey. I'm sorry I've taken a lot of your time this evening, but, and I hope you have it for this evening.
Friends, there's such a good talk, educated ourselves. Satyajit spoke on quite eloquently on the new language for biology this century. Are we equipped to speak? We have understood the enormity of this. Thank you. Professor Satyajit has taken nearly 2 hours to reach this place without the chaotic Bangalore traffic. So we heartily thank him, but in spite of all these things, he could make it today and educate us. Thank you very much. And tomorrow there will be a talk by Professor Maya, or Maya's beverage and foods, of adulteration in food and its prevention. Now the house is open for some questions.
I'd like to go back to some of the things that you showed at the beginning. One of them is we talked about how the ratio of basic to applied to technology development. You showed 3 of them, and Korea seemed to be at the opposite pole from India. But perhaps it's a bit misleading because of the size of the Korean R&D pie. So just by looking at the thing, I got the impression that I'm making lots of approximations, that if Korea spends 5 times as much as India on basic R&D, they can probably spend 50 times as much on technology, and it wouldn't really make any difference. Now, the reason that you didn't put Israel in this chart, it would be very interesting. The reason that I'm asking you this is, because Korea started from the opposite pole, as it were. Virtually nothing on basic R&D. It started with
everything being done too. Yeah.
It started with doing things like making steel and building ships. Now, of course, it's moved much further, whether it's physics or biology or anything else. And Israel would have been interested in the same thing, whether technology development is driven by weapon systems development and things like that. This is not to say that Israel has not been doing basic research. It's been doing that since 1950, if not even earlier. And the reason that I'm asking you this is, in India, how do we approach this? I mean, is it the Homi Bhabha, Obaid Siddiqui kind of thing, saying that, look, my proximity to Nehru is such that I can do these things and then drive the other stuff? Or should we start from the other pole? And I don't know whether you can answer this, but this is a question worth asking. And especially if you look at your last slide, where you talked about whenever Bush is giving equal importance to the social studies and choosing social studies rather than social sciences, and the humanities is important in science policy as well. If I may ask another question, you talked about your collaboration between Nimhans and you talked about psychiatric diseases, but in terms of consanguinity, you earlier said that mental diseases like dementia, etc., might be a result of consanguinity. We had a talk here a few years ago by someone who talked about Alzheimer's. He gave some statistics. After the thing, I went up to him and said, what are these statistics? Are they American? He said, yes. We don't have any Indian statistics. These are two open questions, if you like. I can ask you many other questions, but I won't take up too much.
Right. Yeah, so the first part of your first question, I mean, which was, pertaining to this distribution, and at which end do we begin? Now, I must say that we're not beginning the science technology enterprise in India. We are at a certain stage, and in fact, the distribution that we have in India is this today. Well, yeah, I mean, it is extremely, well distributed. So it's so I think if one could imagine this growing equitably, because there's so much room to grow without even making a dent in our economy. I mean, for all three areas to grow equitably. So any country would like to reach here. I mean, that's the sort of the golden distribution of being able to distribute resources. Maybe basic research would be somewhat less than these two, but I mean that's what it is right now. The basic is a quarter, and these two really the rest.
And so the question I was asking is, moving from less than 1% of
GDP
to something like 3%, which
Yes.
South Korea and Israel might be spending.
Yeah.
What is the path to take? Is my question.
Right.
The desirability of going from less than 1% to something considerably greater.